Good morning and good afternoon, everyone. My name is Mahalea Marshall. I'm with the IQ Consortium Secretariat. I'd like to thank you for joining this morning's webinar entitled Utilization of OATP1B Biomarker forgive me, to guide drug-drug interaction risk assessment. This webinar is presented by the OATP Biomarkers Working Group and is sponsored by the IQ Translational ADME Sciences Leadership Group. I'd like to now introduce today's speakers. Dr. Ryota Kikuchi is a Senior Principal Research Scientist for Quantitative, Translational, and ADME Sciences at AbbVie. Dr. Kikuchi received his PhD in Pharmaceutical Sciences from the University of Tokyo in 2007. After spending 2.5 years as a postdoc, he joined AbbVie in 2009. Currently, he leads the Translational PK and DDI Sciences team within QTAS. His team is responsible for human PK and DDI predictions of small molecule drug candidates from early to late stage in drug discovery across all therapeutic areas. Dr. Kikuchi has almost 20 years of experience in drug transporter sciences. Besides serving as an SME of human PK, DDI prediction, and drug transporters, he has over 10 years experience as a DMPK representative supporting both discovery and development projects. Our next speaker is Dr. Bridget Morse with Eli Lilly. Dr. Morse is currently a Senior Director in Drug Disposition at Eli Lilly Research Laboratories. She received her PharmD from Butler University and her PhD in Pharmaceutical Sciences from the University at Buffalo. Since 2014, Bridget has served in pharmaceutical industry as a subject matter expert in pharmacokinetics, transporters, and drug-drug interactions, particularly in the use of pharmacokinetic modeling for substrates of hepatic transporters. She is actively involved in many cross-functional committees in these subject matter areas. She is currently chair of the IQ OATP1B Biomarkers Working Group and the chair of the AAPS Drug Transporter Community. She serves as a peer reviewer for multiple publications, sits on the editorial advisory board for the AAPS journal. She has over 30 published journal articles and book chapters. With that, I'd like to go ahead and turn things over to Dr. Morse.
So as Maha has introduced, today we're going to share with you the work that we have completed in the IQ OATP1B Biomarker Working Group, evaluating the OATP1B Biomarker coproporphyrin to guide drug-drug interaction risk assessment during drug development. Next slide. Just a quick disclosure that this presentation was developed with the support of IQ, which is a not-for-profit organization of pharmaceutical companies with a mission of advancing science and technology to augment the capability of member companies to develop transformational solutions that benefit patients, regulators, and the broader research and development community. Next slide. Firstly, we just want to acknowledge the contributors to this work, specifically the members of the Biomarker Working Group, Ryota and myself as the co-chairs, Marhelene Meehan from AbbVie as our sponsor, and every single one of the members on our working group. We've had this working group going for over 3 years, and our members have been dedicated the entire time to achieve the output that we're going to present to you today. And of course, thanks to IQ, TELG, and the Secretariat for helping us with the work and with organizing this webinar today. Next slide. Okay, and then first we'll just give you an introduction to the background, the problem statement, and the mission of our working group. Next slide. So this working group was put together to address a couple scientific issues regarding OATP1B-mediated DDIs. And the first is that tools for predicting clinical OATP1B-mediated DDIs using in vitro OATP1B inhibition data are limited. So the first step in evaluating clinical risk of OATP1B inhibition by a potential perpetrator is the use of static models, or R values, and these are used primarily to prevent false negatives and therefore innately result in false positives. So if we look at the graph on the right, this is a publication put forth by members of the FDA evaluating various cutoffs for the inhibitor maximum concentration over the IC50 at OATP1B. And as you can see here, so if we assume that a positive drug interaction is an AUC ratio of 1.25, which is on the x-axis, and then the inhibitor concentration over the IC50 on the y-axis. And so the current recommendation for that cutoff on the y-axis is 0.1. That's the current recommendation by both the FDA and the PMDA, as well as the ICH DDI guidance that is currently in draft. And so if we look at that cutoff for predicting drug interactions that result in an AUC ratio greater than 1.1, all of those in that left upper hand quadrant would be considered false positives. And so that was 22% in this particular data set. And so there's a significant number of false positives, but again, that is the purpose of a static model is to prevent false negatives, so we will result in false positives. And I also want to point out from that graph that OATP-mediated drug-drug interactions can be quite large. Even due to just primarily OATP inhibition, they can be greater than 5-fold for certain substrates, particularly statins. And so that is certainly outside of their therapeutic window. And so while many transporter-mediated drug interactions are not large in magnitude, this is certainly one by which we could get some clinically relevant drug interactions through just this primary mechanism. And then just focusing on the second bullet here, after we use the R value, unlike our models for CYP enzymes, for OATP1B-mediated DDIs, methods to further refine positive R values are lacking. So there are uncertainties in the IBE using mechanistic modeling, and there's a few reasons why that might be, and we haven't been able to pinpoint exactly what that is, but including uncertainties in the in vitro measurements, including non-specific binding mechanisms, including some time dependence, the assumptions in our PBPK models, as well as understanding the clinical contribution of OATP to these rather complex substrates, and we'll touch on that in the next slide. And so this is the first opportunity for the use of in vivo biomarker for OATP1B-mediated DDIs. This is to alleviate dependence on in vitro data for which we have little quantitative confidence and allow refinement of static DDI predictions. Next slide. And then, as I just alluded to, another scientific issue is that drug interactions for our clinical OATP1B substrates may not be due exclusively to OATP1B. So in this table, we've outlined the disposition as well as the drug interactions with what we consider our most selective clinical OATP inhibitor, which is single-dose rifampin, for the statins that are commonly used. And so these are by far the most highly prescribed OATP substrates for which OATP-mediated DDIs have been demonstrated to be clinically relevant. And as you can see here, all of the statins, although they are all OATP1B substrates, they are substrates of other transporters and enzymes. And the transporters and enzymes that we've shown in red are the ones that have shown in vivo relevance using either inhibition with specific perpetrators or pharmacogenetics. And so we have very heterogeneous group of compounds for which we are attempting to understand drug interactions. And then if you look at the bottom row in this table, the drug interactions with the same dose of our OATP clinical inhibitor rifampin show different AUC ratios, all the way from down to 2.3 for pravastatin to over 10 for atorvastatin. And so the static model cutoffs have been established with these OATP substrates of different complexity. So it's difficult to attribute clinical DDIs to OATP1B alone, and therefore we may be overestimating 1B contribution. They also have different sensitivity, again, that the clinical inhibitors, they affect these substrates to varying degrees, and the static models are currently substrate nonspecific. And so another opportunity for the use of a biomarker is that it may improve understanding of that OATP1B contribution to DDIs and also potentially the predictive models if we can better understand that contribution for our clinical substrates. Next slide. And this slide just depicts a recent publication that clearly outlines what the complexity of our substrates, specifically for rosuvastatin. So in this paper, the authors attempted to differentiate drug interaction mechanisms underlying rosuvastatin, clinically observed drug interactions, as rosuvastatin is a substrate of OATP1B as well as BCRP, both of which have clear demonstration of the role in the in vivo PK of rosuvastatin. And so they looked at the hepatic OATP1B inhibition cutoff, again, the R value that we just described using a cutoff of 1.1 as the regulatory cutoff, and they also looked at a value of 1.5, as you can see there in that graph, and then also considered gut BCRP and inhibition cutoff using the current guidance as well, which is currently 10, and they also looked at potentially using a higher value for that. And then if we look at the graph in the middle, this is color-coded by the magnitude of rosuvastatin AUCs. So green being no DDIs, and then all the way up to red, which are large DDIs. And so their conclusion, if we look at, for many of these compounds for which there are drug interactions either in the orange or red coded, they actually have a much higher BCRP inhibition index in the gut, in that their IC50, their calculated gut concentration is over 1,000-fold. If we look on the y-axis, on the x-axis there, over 1,000-fold the IC50 that we've determined, that was determined in vitro. However, really only a couple of compounds, rifampin and cyclosporin, have R values that are much, much greater than 1 or even greater than 2 that have significant DDIs. And so their conclusion from this is that many rosuvastatin DDIs can be explained almost primarily or completely by BCRP inhibition alone, even though many of these compounds did show OATP inhibition in vitro. They attribute the in vivo effect on rosuvastatin primarily to BCRP. So this just kind of outlines that we see OATP inhibition and BCRP inhibition in vitro for many compounds. However, without the use of a biomarker, it can be difficult to explain which mechanism is primary. And this also comes to the point that, again, these substrates that we're looking at are complicated. They can have drug interactions due to other mechanisms than OATP. And our biomarkers, if they're selective for OATP1B, should specifically identify OATP1B-mediated DDIs. It would not be fair to assess it against DDIs, for example, that we think are primarily involving BCRP inhibition. And so, therefore, a critical step in the evaluation of our biomarker was identifying clinical drug interactions that are primarily OATP1B mediated to fairly address the biomarker utility. Next slide. And so then just a couple slides regarding the selectivity and sensitivity of coproporphyrin-I. So by far the most clinical data for any of the OATP biomarkers. There are, of course, several that have been reported in the clinic, but we have the most clinical data regarding the selectivity and sensitivity of coproporphyrin-I as a biomarker for OATP1B activity. So with regards to its selectivity, we do have data in subjects with Rotor syndrome, which is complete deficiency of OATP1B and 1B3, which is what we call 1B. So in these subjects, this was demonstrated all the way back in the 70s. They have significantly increased urinary coproporphyrin-I compared to controls, so indicating that hepatic clearance is decreased and that the drug is being cleared primarily into the urine in these people that have no functional OATP1B. We do also demonstrate a similar but much less magnitude effect of subjects that have MRP2 deficiency. And so coproporphyrin if we look at the bullet points at the bottom, it is a porphyrin metabolite from heme synthesis, and it is a substrate of OATPs as well as MRPs. But you can see the extent of urinary excretion compared in those with OATP1B deficiency compared with MRP2 deficiency was much greater, indicating that 1B transport is the rate-limiting step for hepatic clearance for coproporphyrin-I. And then if we look on the right here, this is a study, much more recent study in over 300 volunteers published in 2021, looking at the relationship between SLCO1B1, so the gene that encodes OATP1B1 transporter, looking at those genotypes all the way from those increased to decreased activity, as you can see as labeled there on the x-axis, And so you can see a clear relationship that compared to reference, those with decreased activity particularly, you can see increased exposure, baseline exposure to CP1, and this has been confirmed in a few other studies as well. So pretty definitive clinical data regarding the selectivity of coproporphyrin-I for OATP1B activity in vivo. Next slide. And then we have also some recent data regarding the sensitivity of coproporphyrin-I as a biomarker for OATP1B activity. And so in this particular paper, we selected this example because it was a nice demonstration of the dose dependence due to selective OATP inhibition due, again, to our clinical OATP inhibitor, rifampin. And so looking at the graph on the left is the increase in coproporphyrin concentrations following rifampin doses from 150 to 600, and you can see a clear, nice dose-dependent effect with our clinical inhibitor. They also assessed the doses of those rifampin doses on 4 clinically used OATP1B substrates and saw dose dependence for those as well. And then they looked at the correlation between those OATP1B or statin DDIs with the exposure in coproporphyrin-I, and you can see there, particularly for atorvastatin and valsartan, which are our sensitive OATP substrates, the correlation was very close to 1, as we've highlighted there at that box, with coproporphyrin in relation to those statins. So pretty good clinical data regarding the sensitivity with regard to our clinical substrates as well. Next slide. And so the initial data, I would say, with correlating coproporphyrin with a change in a drug interaction came out in 2016. And so we have been, the industry has definitely been following this data for quite some time now. And so it has actually made its way into some decision trees. On this slide are decision trees from the International Transporter Consortium, the ITC, as well as the FDA. And in both of these, they indicate the use of biomarkers following a positive R value to guide risk assessment. The knowledge gap that was acknowledged particularly by the FDA, which was just published last year, is what is a relevant change in CP1 exposure that has been associated with a relevant DDI? And so that was the question that we sought to address with this biomarker working group. In order to do this, it requires a robust assessment of change in CP1 exposure and magnitude of OATP1B clinical DDIs across a range of OATP1B inhibitors and substrates. And so that is what we sought out to do, is to further define these decision trees with magnitude of change in CP1 exposure that would be appropriate to use in drug development. Next slide. And so these are the objectives that we set forth for our working group, primarily to evaluate the use of endogenous OATP1B biomarkers, primarily coproporphyrin-I, for refining risk of clinically relevant OATP1B-mediated DDIs, to inform prioritizing, delaying, or replacing drug interaction studies with clinical OATP1B substrate, so a formal substrate or a formal drug interaction study with a statin, for example. And we felt that if we could use it for this mechanism or for that purpose, we should also be able to use it to differentiate mechanisms underlining clinical DDIs when we do need to run those clinical DDI studies. And so we sought to do this through the process of identifying DDIs primarily due to OATP1B inhibition, so not primarily through other mechanisms such as 3A, 4 inhibition or BCRP inhibition, and also by establishing the magnitude of CP1 exposure change associated with positive DDIs, so anything with an AUC ratio for a clinical substrate greater than 1.25, but also associated with moderate strong DDIs. And so for most of our clinical OATP1B substrates, the therapeutic window is considered to be about 2-fold. And so we thought it would be important that if we could not rule out a positive DDI, if we could at least conclude that the drug interaction is not expected to be moderate or strong, that should still help us with decision-making in drug development, particularly when other mechanisms have been ruled out that may affect the disposition of those substrates. And then secondarily, we sought to provide recommendations for the collection and interpretation of OATP1B biomarker, again, primarily CP1 in clinical studies, and identify any knowledge gaps for the application of this data. Next slide. And so this is just an overview of our workflow. So in order to achieve our objectives, first we compiled OATP1B in vitro, clinical OATP1B substrate, primarily statin drug interaction studies, and clinical biomarker data. And this included literature as well as proprietary compounds. Our proprietary data in our final data set was actually limited to baseline values. And so that was all blinded by the IQ consortium. The literature data, we were able to disclose the compounds for each of those. And this also included not just in vitro data against OATP1B, but also drug interaction, clinical drug interaction data and in vitro data against other mechanisms. Again, we'll go through this in a couple of slides that are necessary for the interpretation of those positive drug interactions that we see in the clinic with statins and other OATP1B substrates. And so after we gathered that data, we addressed the potential mechanisms using the data I just mentioned to address the contribution of OATP1B to those DDIs and identify drug interactions that are primarily OATP1B mediated, such that it is a fair comparison for our biomarker. And then we used those select drug interactions to evaluate the relationship between those and the biomarker in order to establish a relevant change in CP1 to help guide decision-making. And we did this through evaluating the negative and positive predictivity and the sensitivity and specificity of the biomarker, again, for two different cutoffs of AUC ratio or drug interaction for those clinical substrates. And so we did this, including the AUC ratio of the victim. We evaluated for the biomarker both the AUC ratio and Cmax ratio or change from baseline of whatever was presented or reported in the data that we had. And we did this with all OATP1B substrates as well as by OATP1B substrate, again, given differences in their disposition as well as their sensitivity to OATP1B inhibition. Next slide. And then this really was our intended outcome, again, as I mentioned, to further refine a decision tree for assessing potential in vivo OATP1B DDI risk by identifying that in the orange box there to identify that relevant change in CP1 at a therapeutic perpetrator dose that would warrant further evaluation in the clinic. Next slide. Okay. And so those were our objectives and our general workflow. Now we're going to go through the data we collected and our output. Next slide. Okay. So as I mentioned, the first step in gathering the clinical drug interactions that we thought would be useful for this purpose was identifying drug interactions primarily due to OATP1B inhibition. We did this very rigorously. We considered each inhibitor regimen and substrate drug interaction individually. So considered the inhibitor dose, the inhibitor dosing with regard to the substrate, as well as the disposition involved in that substrate for each of the drug interactions that we had biomarker data. So all of the drug interactions that we included were those for which we had biomarker data at a similar inhibitor dose and regimen in the clinic. And so this included, again, mechanisms involved in the substrate disposition, those I showed previously in that our scientific issue number 2 in that table, and then also the potential effect of the perpetrator on known mechanisms involved in OATP substrate disposition. And so we did this, we considered these stepwise. And so if there are clinical drug interaction data using an index probe substrate for a particular mechanism, so for example, if there were clinical data with our inhibitor regimen against midazolam in the clinic, then we use that to identify whether or not that compound inhibited 3A. If those data were not available in the clinic, then we considered in vitro inhibition constants and static calculations as recommended by the FDA. So in the table below, we've just highlighted some examples of how we did this. So if we look at the top with rifampin, looking at a 600 single dose. So we have a lot of clinical data with that, that perpetrator regimen. Just looking at PGP, we have data with dabigatran and digoxin to show that the inhibition, clinical inhibition of PGP is weak at most. Same for BCRP. We have data that single dose of rifampin does not significantly affect the AUC of midazolam, so we could conclude no inhibition. And then I'm not going to go through all of those, but if it was green, then we considered it was low risk or no risk, and therefore we could conclude that a single 600 milligram dose of rifampin was going to be primarily OATP1B mediated, those drug interactions that have been observed with that perpetrator. Now, if we look at cyclosporin, this is an example of how we consider the perpetrator regimen. So we have some clinical studies in which cyclosporin was given 1 hour prior and also 3 hours prior to the substrate. And so given at the same time or similar time as the substrate, cyclosporin certainly has a DDI index greater than the 10 that the FDA indicates for PGP and BCRP, indicating that there's potential inhibition. However, when staggered by 3 hours, which is approximately small intestinal transit time, we assume that the drug, that the substrate and the inhibitor would likely not come into, not be in the intestine at the same time. And so that's why there are red boxes for one cyclosporin regimen where there are green boxes for another regimen. And then another good example then is fenebrutinib, which actually has clinical data with midazolam indicating it does affect CYP3A activity, and then also a very high IGUT over IC50 for BCRP, and indicating that their observed rosuvastatin drug interaction probably did involve BCRP. So in order to be included in our primary data set, all of the boxes for these mechanisms involved in any statin drug interaction had to be green. Next slide. Okay, and then this is a list of those studies that we concluded based upon that analysis to be primarily evaluating OATP1B inhibition. We had a nice balanced data set actually at the end of this with 18 negative or weak drug interactions and 16 moderate to strong drug interactions. And I won't go through any of these, but if anyone has any questions on these particularly, we can address them at the end. Next slide. Okay. And then these figures are showing our primary analysis, again, looking at all drug interactions. So all of the data that we have, biomarker data in a drug interaction at a similar inhibitor regimen. And then you can see, looking at the right here, the graph looks quite different when we narrowed it down to drug interactions that were primarily OATP1B mediated. And so we consider this to be our primary output there on the right. And so we were happy that we were still able to include 34 drug interactions in this analysis and in this graph. So there are many substrate inhibitor combinations that had more than one study evaluating the same inhibitor and substrate regimen. And so what we're showing here are the mean of all those studies. Some of them had 8 different reports for the same combination. So the mean and standard deviation from all of those studies is shown here if there are multiple studies reported. And so we had 12 perpetrators at up to 3 different doses in this data set, as well as 6 different OATP1B substrates. And of course, you can see there that it is color-coded by substrate. And then, as I mentioned previously, we evaluated the performance of coproporphyrin-I, if we look on the y-axis there, and the dotted lines that we have for predicting OATP-mediated DDIs with an AUC ratio greater than 1.25 as well as greater than 2, so moderate strong DDIs. And then looking at the vertical dash lines, we looked at various cutoffs for CP1 AUC ratio of 1.25, 1.5, and 2 to predict the OATP-mediated DDIs. And we'll show the table of our calculations for the performance of that in a couple of slides. And so these data are looking at coproporphyrin-I AUC ratio. Next slide. We also looked at the relationship between the drug interaction and CP1 Cmax ratio, again, with the same perpetrator substrate combinations that we had in the previous slide. And if we look on the right hand and consider our primary analysis, those for which we should be measuring the biomarker against, we actually see a really nice relationship between CP1 Cmax and the drug interaction. Such that we could almost, if we look, we could almost bin them into negative, weak, and moderate or strong, just based upon the CP1 Cmax-R. And I do want to point out that in general, the CP1 Cmax-R was greater or equal to the CP1 AUC ratio for all of those inhibitor regimens. Next slide. And so this table is just showing the predictive performance So I'm not going to go through the calculations. You can easily look at our white paper that was just accepted in CPT if you'd like to go through those. But I do want to point out, so if we look at the first three columns here, so for predicting a positive DDI, again, we assessed a different cutoff for CP1 AUC ratio and Cmax ratio. And so just looking at the AUC ratio, so looking at predicting a positive DDI, even using the CP1 cutoff of 1.25, we still had 2 false negatives. Those were from a single dose of cyclosporin 20 milligrams, in which the CP1 AUC ratio was actually 1.23, so barely met the cutoff and was statistically significant, but technically did not meet the 1.25 cutoff that we had. And going to an AUC ratio of 1.5, we had 2 more false negatives using just a CP1 AUC ratio. The sensitivity was still quite high at 92% and 85% for both of those. But ideally, we would have no false negatives using this biomarker data. And so conversely, if we look at use of CP1 Cmax ratio, either a value of 1.25 or 1.5 had 100% sensitivity for predicting positive DDIs. And then if we look at the 3 columns on the left for predicting moderate to strong drug interactions, so we identified 100% sensitivity using a CP1 AUC ratio of 1.5, although if you look here, the specificity is not great at 65% using that AUC ratio for CP1. And then again, as you can see, looking at the bottom left, using a cutoff of 2 for CP1 Cmax ratio, we identified 100% sensitivity, so no false negatives. Again, for identifying these drug interactions using CP1 Cmax ratio, with decent specificity of 72%, so still greater than 70 for that. And so based upon this analysis, we have identified changes in particularly CP1 Cmax that can help us make decisions in drug development. And we have outlined that in a framework on the next slide. Oh, sorry, one more slide. So we also did look at the correlation of CP1 exposure and individual OATP1B substrates evaluated in the same study. So in the previous analysis, even if the biomarker and the drug interaction were not assessed in the same study in subjects, as long as the inhibitor regimen was similar, we combined them. These data show CP1 exposure in the same individual and study for each of the substrates. And so on the x-axis here is CP1 AUC ratio or Cmax ratio in the bottom 4. And then on the y-axis is the AUC ratio for the drug interaction. And so you can see we've got atorvastatin, pitavastatin, rosuvastatin, and valsartan for which we had this data. Using CP1 AUC ratio for all of the compounds except for pitavastatin, there was a significant correlation. But I think it's worth pointing out that the relationship was either close to unity or less than unity, indicating that the substrate is actually more sensitive to OATP inhibition than CP1 AUC ratio. However, if we look at the Cmax ratio for CP1, particularly if you look at atorvastatin, we see a very nice relationship between CP1 Cmax ratio and the drug interaction. And for these, there was a significant correlation for all 4 substrates. And the relationship was either unity or less than unity, indicating that CP1 was at least as sensitive as our clinical OATP substrates for all of the 4 substrates with which we had data. I do want to point out just an important note for this data is that for many, the open symbols in this data set represent single dose of rifampin at one of the 3 doses we had, and so many of this data does come from one inhibitor, but it is the data that we had available. Next slide. Okay, so this is the framework that we were able to come up with based upon the data I just showed in the previous 2 slides. And so, of course, we recommend assessing in vitro transporter inhibition and using a static model that can still be used to conclude low OATP1B DDI risk if it's below the cutoff. And Ryota is going to go through, how this CP1 may actually be helped to refine what that cutoff is. But regardless, we do still believe that the static model should be useful for reducing false negatives. However, if that R value is above the cutoff, we recommend considering the change in CP1 at a therapeutic perpetrator dose. If the CP1 Cmax ratio is less than 1.25, then we consider that low risk if we look at the box on the right for OATP drug interaction. As has been showed in our data set, we have observed no false negatives for positive DDIs when CP1 Cmax ratio is less than 1.25. If it is greater than 1.25, we consider that needing further evaluation, and that the magnitude of CP1 change should be used to guide in vivo risk assessment. And so a couple of questions that can be asked is, what is the change? Is the change in CP1 associated with a weak DDI? So is the Cmax ratio for CP1 less than 2? Again, we had no false negatives for predicting moderate strong DDIs when the Cmax ratio was greater than 2. So we should be able to conclude that if it is less than 2, should not be outside the typical OATP1B substrate therapeutic window. And then additionally, and there are several publications evaluating this, we're not going to go into today, but PBPK modeling can also be used to further refine those in vitro determined Ki values for OATP1B, and then used to predict clinical drug interactions with our OATP1B substrates. And so that can also be performed, and so that can also be a consideration for prioritizing studies. And so depending on the answer to those questions, then we recommend prioritizing formal clinical drug interaction assessment and or inform co-medication dosing in the next phase accordingly. But our general conclusion is that CP1 measurement is recommended in this use of decision tree to inform OATP1B drug interaction potential. Next slide. I think I'm going to hand it over to you now, Ryota.
Yeah, thanks, Bridget. Can everybody see me well and hear me well? Just to confirm.
We can. Yes.
Okay, thank you very much. So, like Bridget nicely explained, what we did so far is by collecting literature information in terms of OATP1B victim DDI together with CP1 changes with the same perpetrator regimen, we were able to demonstrate CP1 is indeed a selective biomarker for OATP1B1 that can be also used early in the clinical development, such as FIH study. What we also wanted to pursue then is knowing that a CP1 is a selective biomarker for OATP1B1, as for example, exemplified in the figure on the top right here. We wonder, one more to add. We also know there is an OATP1B pharmacogenetics, for example, C521T. We also confirm the impact of OATP1B polymorphism is similar between OATP1B substrate or CP1, as you may see in the figure at the bottom right. With this data, we are convinced CP1 is indeed a selective biomarker for OATP1B. Then we wanted to pursue a possibility. Can we use CP1 to further refine the methodology of OATP1B static DDI prediction, such as R-value calculation? With that in mind, what we did is with each inhibitor regimen or perpetrator regimen or non-inhibitor regimen we were able to collect the data for, we evaluated the correlation between OATP1B and static DDI prediction and change in the CP1 exposure, including both AUCR and CP1 Cmax. As you may see in the whole figure on the left, we observe there is a strong correlation between OATP1B1 R value and CP1 AUCR. The other metric we wanted to evaluate was how about simply dividing the unbound maximum plasma concentration for each inhibitor by OATP1B1 in vitro IC50. How does that correlate with change in the CP1 exposure? And here you go. Indeed, both OATP1B1 R value and just the ratio between unbound maximum plasma exposure and OATP1B1 IC50 showed nice correlation with both CP1 AUCR, panel A and B on the left, and CP1 Cmax, C and D on the bar. Then, as you may see in the 3 bullet point on the right, as well as in the table, we wanted to come up with some idea what makes most sense in terms of the cutoff value for either OATP1B1 R value or unbound Cmax over OATP1B1 IC50 ratio. Based on this limited data set, it sounded like OATP1B1 R value greater than 1.5 or unbound Cmax over IC50 ratio greater than 0.1 showed comparable performance in predicting both CP1 AUCR greater than 1.25 and CP1 Cmax R greater than 1.25. This further supported the selectivity of CP1 as an endogenous biomarker of OATP1B. Because we are simply comparing the change in the CP1 and OATP1B IC50 together with inhibitor concentration. With that, we think this indeed suggests the possibility to use CP1 to further refine the model and method to including cutoff value for the static DDI prediction for OATP1B1 and 1B3. Using the table at the bottom right, I just show the performance of each metrics, and like Brigitte said, this data was already published in the CPT, so please go into the paper if you want to learn more. The other thing we wanted to understand is what would be the appropriate study design to collect CP1 exposure information in clinical trial. For example, SAD study in first-in-human trial. In order to answer that question, we first need to try to understand the baseline of CP1. The way we did it, so we just collected a CP1 baseline concentration, AUC ratio in the absence of any inhibitor or in the presence of non-inhibitor. We now compare that CP1 AUC 0 to T or baseline AUC 0 to T with just the product of baseline concentration and last time point. As you may see in the figure on the bottom on the left, indeed, those two values show a very nice correlation. CP1 AUC 0 to T is very close to just the product of baseline concentration multiplied by last time point. This observation is indeed consistent with the observation that with limited study, there is a minimal diurnal change in the CP1 baseline exposure. For example, the one shown on the right. This indicates in the clinical trial, we don't have to necessarily collect CP1 AUC on the baseline. We may simply collect the CP1 exposure pre-dose and then multiply that with our last time point. That may be sufficient to evaluate CP1 AUC ratio and also Cmax ratio. With that, we came up with this conclusion as an IQ OATP1B biomarker working group. First of all, CP1 measurement is recommended to inform our clinical OATP1B DDI potential. The current analysis identified clinically relevant changes in the CP1 exposure, which correlate with OATP1B-mediated DDI. To give some more granularity here, negative CP1 data, such as Cmax ratio less than 1.25, are associated with negative DDI and lack of in vivo OATP1B inhibition. Greater changes in the CP1 exposure warrant further risk assessment. As Brigitte already mentioned, we are able to come up with a number to define clinically relevant change in the CP1 AUC ratio, such as 1.25. The other thing we noted is even if there is a change in the CP1 exposure greater than 1.25, but as long as that number is less than 2, most of the clinical DDI is just weak, which are often not clinically relevant if we consider patient safety. This data indeed suggests CP1 can be used for decision making, for example, to prioritize, delay, and in future potentially replace clinical DDI study using OATP1B probe substrate during drug development. And finally, as I already explained, there is a strong correlation between CP1 exposure changes such as AUC ratio or Cmax ratio and OATP1B static DDI prediction. There is a correlation between R value with CP1 AUCR, where OATP1B1 R value greater than 1.5 results in no false negative prediction with high PPB and high NPB rates. Also, we were able to introduce another metrics in static DDI prediction, which is simply a ratio between unbound maximum plasma exposure of the perpetrator and OATP1B IC50. The ratio greater than 1.5 also showed a strong correlation with CP1 AUC ratio. So that is the end of the story from the 3-year working group. With that, I would like to take any question. And maybe I will go to the Q&A box and see if there are any questions. Okay, I see one question. With MRP2 knockdown in rare disease, is CP1 plasma levels elevated in a similar manner to CP1 urinary levels? So I assume that this corresponds to the data Brigitte showed in the Dubin-Johnson syndrome, MRP knockdown area. Brigitte, would you like to take this one? Or I can also add my thought.
I don't believe we do. I know we don't have plasma concentrations in that study for those subjects, either with Ritter syndrome or Dubin-Johnson syndrome. They only looked at urinary excretion. I don't believe that we do.
I got next question, and maybe I can take this one. When we say IC50, does it mean IC50 unbound always? Great question. The way, so we as an IQ OATP1B biomarker working group collected IC50 data is just we went into the University of Washington Drug Interaction database and pulled out the lowest reported IC50 value. We assume that means unbound, but again, so it could be so dependent on assay condition. We didn't go further how the assay was conducted. Was it done in the presence of protein or not? We didn't do that. We literally took the lowest reported number. Great question, but I'd say this needs further interrogation. Ideally, such number should come from the same lab, using the same cell line, same assay condition. And I am very curious how the correlation would look like if we took that more sort of controlled approach in collecting OATP1B IC50 values. Okay, another question. We tested CP1 for OATP1B1 based DDI evaluation in cancer patients. To be honest, the data were very noisy and I think he meant hard to interpret. Do you have same experiences for using CP1 in various patient population? I can start. At least as far as I know, in AbbVie, we have never determined CP1 exposure in patient population. We are collecting data in healthy subjects during first in human studies. So I don't have any experience commenting on variability in patient population. Would you like to add anything, Bridget, for this one?
Yeah, I think it's something, many companies that are investigating oncology are going to have to deal with, right? Because we don't typically go into healthy volunteers in those studies. I'm not surprised that they're noisy. And I do recall at least two of the data sets, or two studies as part of our data set, were in subjects with in cancer, and we did not include them in our baseline analyses because the baselines were high and variable. So yeah, I think another thing to consider is the power of your study. First in human studies don't have many subjects, and so you may not be powered to find a 2-fold or 1.2-fold change in CP1 just from one dose level. Dose dependence is certainly something that should be considered when evaluating this as well, when you don't think you're going to have a study that's powered given high variability or low number of subjects in those studies. So yeah, that certainly is going to be challenging in certain populations due to the variability. Yeah. But yeah, and we know various populations due to the data with this biomarker now have changes in OATP function as well, right? So, but we still believe that, assuming that healthy volunteers have fully functional OATP, if you evaluate CP in a SAD/MAD in healthy volunteers, it should still be able to help inform the possible extent of OATP inhibition in disease. Other mechanisms might be involved affecting drug disposition, but should still help inform potential OATP inhibition. Yeah. Many layers to that question, but yeah, I'm not surprised, I'd say.
Yeah, but I think that is another area that means more research is required. There are limited data set in terms of CP1 exposure in patient population, but I think we need to learn more as a field. Okay, let's go to the next one. Being an endogenous biomarker, CP1 data may be highly variable in clinical samples. Can you comment on the IQ working group plan on providing some guidance on collecting and analyzing this data? For example, day one or pre-dose baseline, corrected or low values. Maybe I can start. Surprisingly, once we collect the literature data as well as some proprietary information in CP1 baseline value, we did not observe such variability in terms of CP1 baseline concentration. Of course, there are certain variations within the same study, but overall, across more than 10 different studies from different labs, different country, continent, baseline CP1 concentration in healthy subjects seems to be comparable. We included such analysis and outcome in as one of the supplemental figure in our paper, so I'd recommend you go there and take a look. Would you like to add anything, Bridget, on this one?
Just to the guidance on collecting and analyzing this data, I mean, certainly there's no reason why you can't collect pre-dose and post-dose samples in the same subjects to do either an inter or intra-subject comparison. There are reported values, as Ryota indicated, many reported values in the literature for inter-subject variability, and it's low. And that is, also a consideration, not just about the analysis that we showed here, but there's many things that, go into identifying a robust biomarker, including lack of circadian rhythm, low variability. And so, CP1 meets those things, as Ryota pointed out. And so, yeah, I mean, I think our recommendation is the same as for conducting a drug interaction study. Determine what your comparisons are going to be, inter versus inter-subject, to make sure you're powered to define the mean and variability around CP1.
Great. Okay, let's go to the next one. I think this is an easy one. What about CP3? Have the same results being reported with that biomarker? I can start. Indeed, in the limited study, both CP1 and CP3 measures are measured in the same subject. As one of the supplemental figure, we indeed compare the performance between CP1 we compare the change in the CP1 exposure and CP3 exposure in same subject. There was a strong correlation. That's why we thought CP1 is just sufficient. People may add CP3 if that doesn't add additional burden on the bioanalysis, but based on that analysis and without data set, most likely CP1 is just sufficient to define clinical OATP1B inhibition liability. Okay, next one is very similar. What's the recommendation to assess CP1 concentration after either after single dose or steady state concentration of perpetuator? I'd say maybe it's important to get the CP1 exposure after a single dose, but if we anticipate significant accumulation of a perpetuator, it's also worthwhile to measure the CP1 exposure when the perpetuator exposure is at steady state. But if that is the case, I'd still recommend we collecting the CP1 baseline before the first of the perpetuator, because that is a baseline, not necessarily pre-dose at steady state. Anything would you like to add, Bridget, on this one?
Yeah, we addressed this in the white paper too. I think it makes sense if you're going to be dosing, chronically to assess it at steady state. Getting some CP1 measurements in the SAD prior to MAD might help you, narrow down the time points you might use in the MAD. Sometimes in a SAD you can assess higher doses than the MAD, but again, it's up to the investigators, but certainly doing it at the therapeutic regimen is absolutely essential.
Thanks, Bridget. Next question is about, I think, analytical. So what assays are used to measure CP1 concentration and how many different bioanalytical labs were used? My honest answer is I don't know. What I know is in almost all studies, LC-MS is used to measure CP1. But if we are to understand difference in the analytical method, I have to say that is outside scope of this working group.
Yeah, exactly. We combined, literature data from various sources, although I don't think you showed it, Ryota, but you did compare the baseline values reported with those various methods in all those studies, and they were pretty tight. So we don't believe that had a big impact in combining all that data.
Yep, great point. Okay, next question. If I may follow up on the cancer patient question, what would be your suggestion to test early on? What would be your suggestion to test early on in clinical development OATP1B based DDI in patient population? Do you understand? I guess so this person is looking for our recommendation about the stage. At which stage in the clinical development do we measure CP1 exposure in patient population? That's how I interpret. In my mind, as early as possible, especially if that is a cancer subject. And the good thing is, it's not that it's difficult. We will anyway have plasma sample from those clinical trial. So we can just add another analyte in the analysis. That's my, I don't know, opinion.
Yeah, I agree. I don't think the recommendations change any from what we would suggest in healthy volunteers. This biomarker should be used as early as possible to prioritize, or delay the formal study. I think that can be done in either healthy volunteers or the patient population. The data can be useful for that purpose.
Okay. Next question is again about this. I think it's analytical condition. We had a lot of subjects within below LLOQ CP1 levels, causing AUC 0 to T to be much different than baseline AUC, which is calculated, say, from 0 to 24 hours using baseline times 24. Is Cmax ratio more informative of DDI then? In my experiences, I have never encountered a situation where CP1 baseline level is below LLOQ. I don't know, maybe it could be dependent on the analytical condition. But for the latter part of this question, based on analysis and also as Bridget nicely presented, CP1 Cmax out seems to be more sensitive than CP1 AUCR in terms of catching clinical OATP1B DDI. That's my thoughts on this one. So in a sense, the bottom line is most likely CP1 Cmax ratio is more informative in predicting OATP1B DDI. If a person has an issue in getting enough sensitivity in CP1 at baseline, one thing you could consider is why don't you just use reported CP1 baseline concentration? As we already clarified several times, baseline CP1 concentration seems to be relatively comparable across different studies, different subjects. So that could be something you may consider as a sort of preliminary assessment while you're working to improve the analytical sensitivity of your CP1 method.
Yeah, I concur. We've not had that issue with CP1. With CP3, it's certainly an issue, and also another reason why, as Ryota pointed out, we think CP1 is sufficient and probably better for that because it is difficult to measure. But for the CP1, we haven't had that issue. But I would say if you can measure it at baseline and then can't measure it after your perpetrator, doesn't sound like a meaningful OATP inhibition is occurring. So we have to look at the data, but I haven't had that problem.
Okay, thanks, Bridget. And we have two more questions. And we are about time. I think we can go through those two before we end this webinar. Can you comment on light sensitivity of CP1? Can we trust CP1 data if we retrospectively analyze existing samples that were collected without avoiding light? Great question. I don't have a good answer for that. But again, same thing. If there was a light sensitivity, we would have expected, or we would have observed a lot of higher variation in the CP1 baseline between different labs. So for me, it indirectly suggests it's not a big issue. But I don't know whether there has been any good study to specifically answer this question. I don't know whether Bridge has actual experiences on this one.
Yeah, it is certainly light sensitive. So, prospectively, we certainly take that into account. But I don't know of any comparisons in samples that have and have not been light protected and how that affects the results. I think that would be something that would have to be assessed if you're going to use samples that haven't been prospectively collected for this purpose.
And final question. Thank you for your great presentation. You're welcome. And may I ask, genetic variation may play a role in CP1-CPC level variability in individual? The short answer is yes. There has been some reports showing a difference in the CP1 baseline between different OATP1B1 phenotypes. Okay, and do I have more? Sorry, we have two more. Can you comment on the utility of CP1 in characterizing hepatic impairment population published by Pfizer and for PBPK modeling inputs for OATP substrate? Would you like to take this one, Bridget?
Sure. Yeah, I mean, as we mentioned, the identification of CP1 for, identifying the relationship between that and OATP-mediated DDIs in healthy subjects has certainly enabled us to infer, at least semi-quantitatively, what's happening in patient populations by looking at concentrations of the biomarker. That's certainly true. And so I think I would refer to the authors of that publication, but it is a nice, robust publication that I'm sure in the future we will, make sure that it can be reproduced as we're all running those studies. So I think that's what's needed to confirm that. I certainly have some opinions on the PBPK modeling inputs for the OATP substrates. They're very complicated, and I think we just need to acknowledge that when we're building these PBPK models. But as long as, they need to be verified for the purpose of OATP1B inhibition as extensively as possible. And again, they're very different, and, very careful consideration needs to be considered on those substrate models. Yeah, that to me, honestly, is the rate limiting step in doing those prospective evaluations.
Thanks, Bridget. Okay, let's go to the last one. In vitro studies have found substrate-dependent inhibition of OATP1B1. For example, range of IC50 data for same inhibitor. Do you think there are enough clinical data to evaluate whether substrate-dependent inhibition is relevant in vivo, perhaps combined with PBPK model? I will start adding my personal opinion. Yes, we know there is a substrate dependency in OATP1B IC50 value, but in my perspective, a bigger issue here is even if we consider substrate-dependent IC50 value, but oftentimes we still see disconnect between in vivo IC50 and in vitro number. Generally, in vivo IC50 value estimated by PBPK modeling tends to be lower, more potent than what was reported or what was measured in vitro. That's why we think indeed a biomarker such as CP1 could play a huge role here. That could also fill the gap. There might be still less than perfect IBE in OATP1B IC50 value, but with evaluation of CP1 early in the clinical development, most likely that could fill the gap, and that may also lead us to get the real in vivo IC50 estimate. Anything would you like that, Bridget?
Nothing to add to what you said.
Okay, with that, we were able to cover all of the questions posted online. Mayuri, how do we finish this webinar? Sorry, I should have asked you earlier.
That's okay. Yeah, we can go ahead and wrap up.
Okay.
So I'll just go ahead and thank you both for presenting and addressing the questions submitted by the audience. I think it was really well received, and let the audience know that if they have any additional questions, you can certainly send them to me and I'll forward them along to Ryota and to Bridget. I'm sure they'd be happy to take them for you. And with that, I'll go ahead and close the webinar. Thank you very much, everyone, for joining us this morning, this afternoon, wherever you may be. And I hope that we see you again on a future IQ webinar. Thanks very much, everyone. Take care.
Thanks. Bye.
Thank you.